Cuteness.
All right, welcome everyone to RV Falkor. We're out here on the Cairns Seamount off of Queensland, Australia. And with us today is Dr. Brooke from Geoscience Australia and Dhugal Lindsay, who is joining us remotely due to COVID restrictions. And you are watching live video feed from ROV SuBastian.
Thanks very much, John. And hello everyone that's joining us through YouTube and other online media. Yes, now moving through the water column and part of the dive 374 Cairns Seamount. It's the mid-water part of our dive. We're moving through the water column and recording mostly the jellyfish that live in the water column. And this is part of a key element of the research of this project, led by Lindsay at JAMSTEC in Japan. And we have Dhugal participating in the survey remotely through Falkor's satellite communication system. And we're very fortunate to have Dhugal join us live, and he can provide a bit of a background and description of what we're seeing. Over to you, Dhugal, if you can hear us.
Yes, thank you. It looks like we have here in the camera a lobate ctenophore. Other name for ctenophore is a comb jellyfish. It's a different phylum, completely different group to the regular jellyfish, but because it's soft and squishy and mostly transparent, it's called a jellyfish. These comb jellyfish, you've got 8 rows of combs there. You can see them reflecting the light from the ROV. They served like little oars, little paddles, moving the animal through the water. This particular guy looks to be a lobate ctenophore. On the other end of the animal, where the mouth is, opposite where all those glistening combs are, there are these 2 large ear-shaped lobes, which it uses to catch its prey. The internal surface is somewhat like flypaper. So any little planktonic organisms that get stuck on it, they adhere to the inner surface of the lobes and are then carried by cilia to the mouth, or by the tentacles to the mouth. So what we're trying to do here, because these ctenophores are very, very fragile, doing work with an ROV like we're doing now, actually recording them while they're still alive in situ is one of the only ways to characterize the animals, because you try and preserve them in formalin, which is what is normally used for preserving museum specimens. They tend to disintegrate, and you end up with a jar of formalin and go, I'm sure I put an animal in here, where'd it go? So we're trying to get video of these animals to record them now in situ, especially the area at the top, the area opposite the mouth, the top of the stomach, where the canals are branching off to the comb rows, is very important for telling what group within the ctenophores this animal is. So hopefully we'll be seeing that. It seemed very, very flattened, which tells me it's narrows down the group to 1 or 2 families. So we'll see how zoomed in we can get on the other bits we need to see. These animals can prey on very small planktonic particles, and this area we're diving in, far from shore, the production is not as great as in coastal waters. So animals that can live on less food or on smaller food are more likely to be abundant. This is one of the very first in-depth surveys of the waters off Eastern Australia using a remotely operated vehicle. So we're getting new discoveries as we go, as we dive. It's, oh, that's a new record for the Southern Hemisphere. Oh, wow. I mean, it's just coming fast and thick.
So Mrs. Center Screen, Dhugal.
My feed's pretty dark now. I can't actually see anything that's on the screen.
Yeah, we've got another little dinner plate one for you, Dhugal.
Oh, great. So you got 14 to 16 tentacle kind of numbers.
We're doing that count now.
Yeah. Moving around a bit.
All right, I see him, yeah. So we just caught an animal similar to this in one of the suction samplers. Wow, that was a krill. And there's a pteropod, a pelagic snail, which is just swimming past there with that little triangular shell. There he is. These guys, because the shell is made of calcium carbonate, as the ocean becomes more acidic, the shells get thinner and thinner, and people are worried about if they'll be able to survive it. We still have our Solmissus somewhere in the field of view.
I still have
Oh, man. The ctenophore is still there too?
Yeah. Still looking for the...
Can we go back in on that ctenophore if possible?
Gotcha.
I didn't see any oracles on it. It might be an undescribed family. Just need the money shot to tell for sure. Oh, this is a different individual to the one we were just videotaping. Oh, this is a guy we saw two of yesterday. Money shot. Very, very small lobes compared to the rest of the body. And I've lost the images now. So.
Yeah, we've just turned some lights off to see the illumination from that particular specimen.
Okay. Yeah, you can see the coma is reflecting. So we're about 40 meters off the bottom now, aren't we?
We're at 404 meters depth, looking for a target depth of 800 meters on this dive.
Water temperature of 13.4 degrees Celsius.
So if we're able to meet that 800 meter depth, we'll be going through the Antarctic intermediate water, which is flowing north along the coast of Australia. So we might see some plankton that's been swept in with that current from way further down south. All of these little white dots you can see on the screen there are particles of marine snow. Marine snow is a term we have for anything that, well, you see it looks like snow on the screen. It can be the carcasses of dead animals or as they molt, the molts of the animals. It can be what's left after the animals have eaten and excreted, scales, flocks of phytoplankton that stick to each other with the mucus that surrounds them. And all of these things sink down into the deep sea to provide food for the animals that live there. There's quite a lot more marine snow around here than you would normally expect for an oligotrophic area. Oligotrophic meaning not so much food around. There seems to be quite a lot of food. This could be because it's close to the Cairns Seamount, and there may be upwelling here, bringing nutrients up into the surface waters and stimulating primary production. so more plants can grow because there's more nutrients for the plankton to live on. We've seen a large number of pelagic sea squirts, tunicates they're called salps, which are basically semi-transparent barrels that swim through the water, filtering the water as they go on a feeding net. So they filter all the small particles of marine snow out of the water to eat them. And as the water comes out the back end of the animal, it propels the animal forwards. We've seen both solitary zooids of these salps swimming around, and also chains of the asexual reproduced chains of salps. And when these salps are joined together in chains, each one of them propelling themselves forwards, they can reach fairly high speeds and can be quite difficult to catch with an ROV, which you wouldn't expect looking at that little transparent, fragile looking animal. One of the main groups of jellyfish that we're looking at here is something called a Narcomedusa. And the Narcomedusae have evolved to feed on other gelatinous prey. And they do this by a combination of having tentacles that, instead of hanging down from the rim of the bell, the tentacles point forwards in the direction in which the animal is swimming. And this acts like a stealth mechanism, where as the jellyfish swims through the water, it creates a bow wave in front of it, which can alert its prospective prey to the fact that a jellyfish is about to come on it and eat it. So these Narcomedusae hold their tentacles in front of them, and the tentacle will hit the prey jellyfish first before it realizes it's being followed by the rest of the animal. And they are able to sting the prey with these stinging cells called nematocysts. And the Narcomedusae have nematocysts with extremely long threads, barbed threads that when they eject go deep into the jelly of the animal they're trying to prey on. And that way, even if the animal is very, very fragile, some of the barbs along at least some of that length are going to catch in the tissue of the animal, and they're able to bring it around to their mouth to ingest it. So as we fly through the marine snow field in a kickback to the Star Wars movies, it seems, we're looking out for these very transparent looking animals and trying to stop a great big hunk of steel on them before they swim away. Very challenging, the midwater operations. I'm sure the pilots will have oh, here we go. That's a Prayed siphonophore. A siphonophore is a colony of jellyfish polyps. Usually polyps live on rocks or on wood or attached to some kind of a substrate. But the siphonophores are a planktonic group of jellies where the polyps float or swim around just in the water without attaching to anything. Oh, here we go. There's another Solmessus. Let's see if we can get in on here. It's got its tentacles all splayed out to the sides. This is its stationary feeding position. Oh, he knows we're coming. Started to swim away. You'll notice the tentacles are leaving from partway up the surface of the bell rather than from the rim, which is how we tell if it's a Narcomedusa or not. It's a dinner plate jellyfish, Solmessus. Does it have stomach pouches? There are several groups that look very similar, like this. We can tell the genus by whether it has invaginations of the stomach, usually U-shaped or triangular, coming out from that central mouth and stomach and meeting at the base of the tentacles. This animal doesn't seem to have any. See if we can check that. I cannot see any stomach pouches. So the next thing to look for is tracks of nematocysts, lines of stinging cells on the outer surface of the bell around where the tentacles are attached. And that will tell us which of the two genera with no stomach pouches it may be.
Great images.
Yeah. You able to make out if it's got stomach pouches or not? They might be really shallow.
Coming in close now to see. Look at just that, Dougal.
Could be a pagantha.
Want to collect this, Dougal?
I definitely would like to collect this, yes.
Do you want more images, or should we proceed with acquiring it?
Do we have a good side? I think we've got a good side on. Do we have a good front on? Where we can make out the stomach pouches are there or not? Like a top view?
Yeah, I think we got a couple, but...
You got a couple? Yeah, okay, let's sample it then.
Yeah.
We can check if it has nematocyst tracks back in the lab.
Jarod's going for one more good shot.
Okay. There you go, got all the angles there. That's good.
Give the samples.
Oh yeah, nice. Okay. Huh. Holy fricoly. Yes, definitely. Let's take this. The groups with stomach pouches and without stomach pouches are currently put in different families, but genetic information suggests that it might not be as clear-cut as that, and stomach pouches may not be such a valid character. We need more individuals with a range of different morphologies to actually test that, though.
There he goes.
Excellent. One in the bag. Yep, looks to be gone. Take the squidgy in that canister and see if he comes through.
Thank you.
We got him. Thank you very much. Nice piloting, guys. Without ever actually having tried to catch an animal in the mid-water, it's hard to appreciate how difficult it is to be chasing this tiny little animal which is swimming around in three-dimensional space with a great big hunk of metal, which is also moving in three-dimensional space, Plus, you can be pulled by the tether from behind, so you're trying to go in one direction, but you're constrained and can't. So catching these guys is very challenging.
He's in the bottle.
Excellent. Good work.
Happy to continue descent, Dougal?
Yes, thank you. A lot of ketognates today that I didn't really notice yesterday. Ketognates or arrow worms, voracious predators. Don't just eat copepods, but they're also cannibalistic and they eat each other, even if they're the same species.
Lovely.
Yeah.
So we're now on our way back down through the water column to a depth of 800 meters, where we'll begin our benthic component of the survey.
So as we go through the mid-water, you can see the marine snow particles are coming from the distance towards the camera. We're moving the ROV through the water in this way so as every particle is in focus for at least one frame, and we can identify, hopefully, the larger particles, what they are. It's a Medusa, I think, just there. What do we have? Hello, hello. Yeah, I saw something pulsing. Is that a swimming lobate or a bell-shaped Medusa?
Very small.
That may be Thalassocalis, which is a Medusa-shaped ctenophore. Take a good look at him. It's certainly looking that way. Yes, it is. Thalassocalis inconstans. Ha! So this is a ctenophore which has evolved to have a shape that looks like a jellyfish, like a Medusa, a bowl shape. And it's contracting its bell there to swim through the water. When it catches a krill or something inside the bell, it can also close the rim of the bell very tight so the animal cannot escape, and then dangles its tentacles down into the bell cavity to eat them. Very nice. Quite deep too. A Japanese name for this animal is the mask Medusa. It's a side-on view now, but if we get it from above, you'll be able to see it's rounded, and it has what look like eyes on a mask. And these are actually the gonads, the reproductive organs. Ctenophores are hermaphroditic. They're both male and female at the same time, and you only need one ctenophore to have lots and lots of little babies. That was Thalassocalis inconstans. Thalassocalyce. There's a euphausid, a krill, swimming around there as well. So the food is where the predator is. The way it should be. Looks to be another jellyfish there. Maybe another Narcomedusa. Also less than 20 tentacles. Flattened bell. And
Yes. So do you need some good images of this one, Dougal?
yeah, image to see if it's got stomach pouches or not.
And then would you like to proceed with sampling?
If it's got stomach pouches, definitely.
Roger.
Because the last one didn't look to have any. We're still on the first canister, aren't we?
Canister 3, I believe.
Yeah.
Do we already have 2 of these?
That's affirmative, yes.
Okay.
So Dougal, do you need all your samples in separate jars, or can they be combined at all?
If they're a different group, so I can definitely tell the difference, they can be combined. If it's like these guys, they have to be in separate jars to each other. So I know which video is associated with which physical specimen. But if it's like one black jellyfish and one clear jellyfish, they can be in the same jar.
Okay, thanks.
Good shot. Yeah, if you could suck this up, that'd be great.
Pardon? Yeah, separate jar, of course, yeah, Dougal?
Yes. Yes, please. How were you with during the benthic dive for canisters? Did you run out, or did you have enough?
No more space. We're generally filling up our designated space. That's correct.
Okay.
Which is a good thing, but
So we've now got 4 samples.
Okay, oh, you caught him already. Cool.
Yeah. So we've got 3 samples in 3 jars, Dougal. So we've got a total of 7 jars.
Okay. It's 10 jars and 3 of them are full.
7 jars and 3 of them are full.
Each with an
7 and
individual.
3.
Yeah, each with an individual jelly.
Okay, so there are 4 left.
Correct.
Roger that.
Yeah. It's in the jar.
I'll believe you on that. hopefully we'll be able to definitively say now what this shallow morph with fewer tentacles is, which has been eating at me since the early 2000s. We finally got enough specimens.
So Dougal, just giving you an update on our depth. So we're just past 420 meters.
Right.
Our target depth at the moment, with a slightly shorter dive today, is 800 meters. We're keen to get down to that depth. So if you're keen to move on and...
Yes.
Yep.
Yes, please. Is it a very steep slope? Like, is there lots of horizontal distance you have to travel to get to, so we can hit that depth?
Not as bad as the deeper locations. So yesterday we started at 1200 and there's quite a horizontal distance from there up to slopes that you referred to. But if we start at about 800, yeah, the gradient does increase.
Ooh. Is this the halotrephes you caught yesterday? Certainly looks like it. Oh, there's a cast next to the Falkor as well. It's hightailing out of there, isn't it?
He doesn't
There
like
he
it.
is. Yeah, it looks to be halotrephes. This could go on one of the other, in one of the canisters where we already have something in there.
Roger that.
Okay, so you want to collect him, yes?
Yes, please.
Yeah, do you need
The one
more?
from yesterday survived, didn't it?
Yeah, I believe so.
Oh, and there's a prayed siphonophore. Possibly prayer itself. That really long siphonophore also in the image can reach up to 40 meters in length, twice the length of an adult blue whale. Not the kind of specimen that easily fits in a suction sampler canister.
Dougal, just for the ease of Marty and Jeremy in the lab post dive, we're going to put this in a separate jar just so that we're not trying to tease apart two gelatinous bodies. Cool. Yep.
This second specimen should allow us to definitively resurrect the species, I hope. Playing hard to get. Did he go in?
Yep. Be
Awesome.
sure.
Excelente.
Yep.
And that's confirmed.
He's
Yeah,
in bottle.
there it is.
Good.
One. Awesome. Very nice. This is an animal that was described very early 1900s, which a taxonomist after that decided it was the same as a different species that was described before that. But I think this specimen and the one we caught yesterday will allow us to prove that Van Hoffen was actually right, and it is a separate species. So that'll be very nice to look at in the lab later. And to have the images of it alive, in situ, in pristine condition to go with it. The animal is called Halitrephes, Halitrephes. Self-chain, perhaps? Yes, self-chain. Big tuniform? Is that a tuniform? That's another cestum. Venus's girdle is its common name. It's a type of comb jelly. Very elongate. Swims through the water, kind of like a snake, undulating as it swims. It's quite deep, actually, for a Venus girdle. At 100 meters deeper than I've seen them before. It'd be interesting to have a look at what the salinity and temperature are doing.
Temperature's 11.9 degrees Celsius.
Still very warm, right? Okay. So they like the warm water. Not that worried about the depth, maybe. Oh, what do we got here?
Yeah. Is it a tuniform?
Is that one of those thalassocalis that we just had before?
Yeah.
Right. Just keep heading down. Oh, what's that?
Is it a fragment, Dhugal?
Oh, it's salps. It's salps. That's okay. Keep going. Yeah, it's three asexually reproduced salps in a chain. Something big must have swum past and broke up the whole chain and just left that little fragment.
Yeah.
So we're at depths where we're likely to start seeing suggested shrimp kicking in. which will be probably red around the head and clear or white around the tail. You can tell they're suggested from other shrimps by the antennae bend suddenly, like an elbow bend, so that the antennae lie along the same axis as the shrimp's body. And it's kind of like if a fish comes to eat it from the side, it can sense the fish using those antennae before it reaches the body and is able to escape. Was this a tinophore, perhaps? No, it looks like a discarded larvation house. Maybe an inner filter. Could be Bathycordius too. This is the filtering feeding structure of a pelagic sea squirt. Yeah, double lobe structure. Okay, thank you. Very nice. That's a lot of good food if one of those big particles lands near you and you're a benthic animal sitting on the mud.
Party time.
Or an urchin on a slope.
Ooh, lots of, are they krill? Lots of little crustaceans, I think we just went through. Solitary salps, like the salper, it looks like. Oh, there's something. Big tinophore, it looks like. The lower one. Oh, do we have Kirhimea? Oh, it looks a lot like it. Let's see if this has those filaments coming off the triangular projections at the top or not. My money's on that it doesn't have them. Oh, wow. If this is Kirhimea, it's the second record for the Southern Hemisphere. The first one being a survey of Osprey Reef we did with Picasso 10 years ago. It's a nice big animal, it looks like, fully mature. Just got to see it from the side-on angle.
Yeah, it's very complex.
Yeah, it's facing us now. The mouth is pointing towards us, and the lobes are out, like around the mouth. It's got 4 large ribbon-like structures, which are called auricles, and it moves them through the water and stimulates copepods and things to escape from this sudden movement of water. And when they flip back to escape the auricles, they land on the lobes, and they get hit by that flypaper-like sticky structures called coloblasts there. And then it'll gradually move the animals it's caught to the mouth and eat them. me see if that has filaments or not yet. It looks like it doesn't. Yes, that's a Kiyohimea. Kiyohimea usagi. Kyohimea, which means the pure princess in Japanese. Usagi, Usagi means rabbit, because of those little rabbit ears pointing out the top of it. Actually, the bum of it.
I don't quite see it, but yeah, nice name. Beautiful.
Yeah, and hopefully we'll have a size with that by the GoPro cameras.
What sort of estimate size, Google?
It's a big one with non-submersible ones out the window, because normally you can't tell with an ROV how big they are.
Yeah,
but it was a meter, maybe.
Wow.
Try fitting that in a sample jar.
Indeed.
Okay. Let's keep going down.
Is that enough? Yeah, we'll keep going.
Yep. I'm amazed at the depths that some of these guys, some of them are deeper than what I'm used to, like the Cestum just before. So it must like the warmer waters and can go deeper. But that Kiyohimea, for example, is at the same depth I'd expect to see it off Japan, even though the water characteristics, the water mass characteristics are so different between Japan and where you are now.
Yeah, it's still 9.6 degrees Celsius here at 497 meters.
Right. Wow. We're trying to do some habitat mapping to see exactly what it is that drives the distribution of these animals. So being able to get good quality data like this at a species level from off the coast of Australia is extremely exciting. What's the oxygen level like right now?
Good question. Do we have that up? .64. 230.64.
230.64, did you say?
Yep.
230. Have to convert that to milliliters per liter. Oh, another ctenophore. Might be a little bit dark. Oh, that's a self chain. They're hightailing it. Each one of those white spots is a stomach full of food. And you see, because they've been, it's Selpa, the genus Selpa. That white pointy bit on the front of the colony is roadkill. As it swims through the water, it's collected all that marine snow, like you collect insects on your windshield. And as the marine snow flows along both sides of the animals, they suck them in and filter them. the brownish tinge to the guts there is the partially digested chlorophyll phytoplankton material. Moving down. Is something? Pyrocene, perhaps? Center screen slightly left.
It's gone. We're just changing ROV pilots, Dhugal. Chris on board now.
All right. Roger that.
Was there something on the left, Dhugal? Sorry.
Yes, straight ahead of you, slightly to the left, there's a solid white object, possibly a pyrocene. Is that him? I think that might be him. That's not a pyrocene. No, no. That's okay, thank you.
Shall we keep diving?
Yep, keep diving. Is the screen slightly darker than it was before?
I think so. How's that?
No, yeah, that's great. That might be the pyrocene. Oh, schema?
Then coming up on the right.
Oh, Bathysaroe. Film this for a little while, please.
Once this one, if we can. Yeah, that's it.
Looks like it might be the same species as yesterday. And then that other lobate with the small lobes as well. Wow.
You just want images of these, Dhugal, or should we collect?
No, that's cool. Just drive through him and go to the one that's further away. It's good. Yep,
yeah, no, let's see what this space.
It's very small,
whatever it is.
He's tiny, they're saying.
Is he? Right, okay. Whoops. Looks like you cut through the logo. You may grow back. There might be two of them next time. Okay, let's keep going deeper then. Oh, look at all the krill. Wow. Krill everywhere. They might be attracted to our lights. That's a lot of food. Wow, they're very deep. Oh, that looks like a pyrosome, perhaps. Going close. There's something attached to it. That's the seafloor.
Dougal, we're starting to get a bit short on time, so we might get going a little bit quicker to our waypoint, and that's where we'd like to start the benthic part of the.
Roger that.
Hey Google, we're basically going to turn to the south here and start onto the. So I'm going to get a pause there, and once we pass it, I'll just start bringing you after this west there. That's cool, so cautious. And then you got your head there, or how's it going?
Dhugal. We've got a bit of a change in plan to fit everything into our schedule. We're at a predetermined waypoint about now for a 500-metre transect, following the operating procedures for that work. So yeah, we'd very much welcome if you'd like to stay and contribute to commentary. That would be great. Also be Jeremy Horowitz shortly.
Roger that. This is very interesting. Rugosity, or like the seafloor. Is this what's going with that texture?
Yeah, it is interesting. I think what we're looking at is a whole series of slope failures of material coming off the sides of the mount, the sides of the reef system that's formed on the seamount, and those deposits have also been eroded, and some of them have been, more cemented than others. So I think it's a continual cycle of material coming off the side of the seamount.
Right.
Some of it being cemented and lithified, and other bits of that being subsequently eroded again further downslope.
And there's a light dusty sediment on top of everything there?
Yes, indeed. So I think that's a annual process of sediment accumulating slowly, but also there must be these episodes where this coarser material is being stabilised downslope quite rapidly due to failure of the flanks of the seamount.
A few years old just before.
Stony coral colony. So I'll now hand over to Jeremy Horowitz, who's our deepwater coral expert.
Hi everyone. Okay. We've got a few hours to collect biology and rocks, so we're going to try to make the most of it. Primarily, we're going to try to get shallower. But as we're doing this 500-meter transect, we saw coral, so we just want to take quickly, probably with a fragment. Look close. Okay, so there's an azuzendellate coral. See some of the tentacles out. Looks like it's feeding. Cool. Alright, so we're going to grab, oh, you can see a little crab on the inside of this coral. Alright, so we're going to grab a fragment of this colony, then we'll continue our transect. So. Yeah. Yeah, like one of those fragments. One of those branches. Yeah, like this one. Thanks. It'll break easily, so beautiful. Perfect. You can put it in the easiest box. Okay. Hi, Noonie. Crustaceans don't explode. We actually often find when we collect corals that at the time don't realize have associated critters. They're often alive even when they get to the surface. So, no, they can travel from thousands of meters deep up to the surface and be alive and well. Yeah, so we can continue our transect. So we're going to try our best not to stop too much. We're going to try to get this transect completed. And afterwards we'll have some time to explore. Ideally we want to try to collect some of the shallower corals, the top of the seamount. But the ROV is a lot better when the water's colder. So we'll probably have to do a bit of rising, collecting, and then dropping back to the cool. But we'll be in the midst of the action, you know, in the top % of the seamount. Looks like more coral, brittle star. More brittle stars on the left. Or these yellow brittle stars. Yeah, those look pretty good. Ooh, that does look good. But try to get to the top.
Hello, Adam.
See if there's an issue.
Oh, hey, Eric.
Relax now. It's me up here now.
Oh, Eric, move the boat. Yeah, we're gonna start moving a transect.
Okay. Just throwing it out there.
Hey guys, we're just switching our sound system. Do you guys all hear me all right? Just let me know on YouTube and Facebook.
I'll speed him up a little bit so you're not stretched way out there.
I appreciate it.
Thanks. Okay, we're seeing more brittle stars and hard corals. So we're down at 477 meters.
Nice transect you're doing.
Yeah, beautiful.
Transecting machine.
Did you see that line? Transecting your division.
I'm gonna cross it.
The sound is better? Okay, good.
Cross that line for line. That's
Transect's a hard pun word.
yeah.
Oh, did you see that fish?
Questions on that?
It's okay.
Yeah, sure. Anything else you got?
Then you can see
That's
both
like-
on the Facebook.
I have it this.
Fishy. Go down or go up on your. What you got? No, it's going to plot the weightiness. So either the
Yeah.
shutter
It's
speed or
it's
light.
It's the
Light on. Yeah. Yeah.
I remember when that's when we turned it on.
Turn it intercast. Open? Yep. No shadow. Then open up your eyes and touch.
That's on holiday. Yeah.
I think so. Maybe your eyes isn't so color.
How come we don't see fish? Well, there are certainly fish around at this depth. Obviously not an abundance of fish. But as we get shallower, we're going to see a lot more fish. At the top of this seamount, there's going to be a lot of fish, hopefully sharks. There's a lot of activity going on at the upper portion of seamounts like this one. But, you know, we see the singular fish swimming around, pelagic fish swimming by these depths. This side of the seamount seems a little bit more bare. It's really steep, and so sediment will fall down the seamount and make it challenging for anything to grow and survive. But once we get a little higher, similar to what happened yesterday, once we hit, I guess about half the depth that we're at now, we start seeing an exuberance of life. And so we're on the other side of the seamount now, and so we expect to see similar compositional shifts from this to algae and fish and high diversity of coral at mesophotic depths.
They're comparable.
Comparable to what?
All the other clubs. They're the green. Except for nibs. It's not the case.
I think my next year matching electrical sometime around then.
Nibs, I think they are.
So yesterday we collected mostly deep corals, and so we're going to try to target some of the shallower corals today. Notably, we're going to try to collect some aquapora, hard corals. The taxonomy of aquapora, we're starting to learn with new molecular techniques that what we thought we knew was actually mostly incorrect. So with next generation sequencing methods, we're learning that the taxonomic boundaries aren't yet fully understood, but they're becoming clear. So now that we know this, we're trying to sample hard corals in the aquapora of all the different species, and then we could then rewrite essentially the taxonomy of the group. So it's really important here that we collect these aquaporas from this Cairns Seamount, which have, it seemed yesterday that we had a high diversity of hard corals. So we could very quickly collect a few, and they'll directly help us understand, better understand how these different hard coral species are related, and essentially what is a species, and where are those species boundaries, what are the morphological features that separate different species. Yeah, and similar to yesterday, we saw this urchin-dominated habitat. Pretty spectacular. See some fishing line. Again, similar to yesterday, this is a really popular fishing location. So, it's not a surprise that we see some sort of anthropogenic impacts. Okay, see another hard coral.
Yeah, 12:30 this morning. I woken up with this weird noise. No. What is that?
Oh, that's cool.
Well, that's a fish.
Nah.
That's a lure. That's a lure.
Oh, it's lure.
Oh, see, we were even fooled by it. That's a good lure.
Yeah.
No, the clock on my wall started freaking out and started progressing it went from 12:30 to 2 o'clock in the morning.
So straighter to the Amityville horror.
Yeah. So I tried to rip it off the wall and throw it in the.
It's your neck of the woods, isn't it?
Amityville, yeah. Oh yeah. I've actually been to the house when I was in high school. Yeah, drive by it.
How old are you?
32.
I was stepping in. I don't even know our mom was last night.
Oh yeah, I know her.
She's like the same age as Long Island.
In the United States, that only works in Canada.
In grade 8. Yeah, even you can actually don't know what literally 3 years.
Well, one of the there's not a lot of jagged.
So our resident sea turtle expert, Christina Bahang, says that leatherbacks can actually be found during these dives. So they can dive to up to 1,000 meters. So I don't think it's very common for ROV dives to see the sea turtles swimming by, but something to look out for.
We see encompasses Queens County, Nassau. You would know better than I would.
Quite great.
Anyway, so I tried to rip it off the wall to go in the hallway, but it was bolted down. So I just opened up the glass part and held it in place until it finally died.
Really bothered you, huh?
Huh?
It really bothered you.
It was like that loud. It woke me up with that sleep.
Could we pause at this depth, at this altitude for 1 second? Someone's asking about the length of the size of these urchins, so we can quickly just throw the lasers on.
I can come down and touch and lasers. Look down, way up, put it there.
Look down? Yeah. All right, we're going to check the size of these sea urchins.
They're just to the right of the
Yeah. So that's 10 centimeters. So that large one has spines that are maybe 3 or 4 centimeters long. But those smaller sea urchins with larger spines, if we could just pan up. So there seems to be 2 types, with my very limited knowledge about sea urchin taxonomy, that the spines of the smaller sea urchins may be 8 centimeters, give or take, 2 centimeters. Yeah. Okay. Asking.
We're going to leave the lasers on. Do you care?
Yeah, might as well leave them on. Sure, it's actually useful. So we're now at 420 meters. The temperature is 12.2 degrees Celsius. So getting a little warmer.
We're going to hit a spot where it's going to jump. Yeah. On where it goes. Several degrees and stroke. So Nancy. Where's our image? There it is.
So there hasn't been much of a change in the last 100 meters. But as we get shallower, we'll see distinct changes in the habitat and the assemblages of biology. I guess these are long-spined sea urchins.
Yeah. Something like that. But it makes me wonder whether it's crystalline rock. It's breaking apart. I'd like to find out. We'd like to try and get a rock soon.
Yeah. Should we just go right there where there seems to be some sort of a breakage?
Yeah, we would like to try right off of this. It's not that 6 minutes. So if you see it, then maybe
Still the stuff sticking out right there. I got faith in that.
Yeah.
What about up there? You think up there over here?
For real quick, because this stuff is so hard that we could sit here all day. So if we see it could be like no weakness now.
Yeah. You think you can work with this now right here?
But if it's really hard, just keep going.
And the stuff we tried yesterday was like rock.
But we'd like to get, from this sort of smooth area that looks like it's a different type of rock.
So somewhere in this area, okay.
Yes. There was also those canyons we went over before.
But yesterday,
Yeah,
the bridges. Yeah, we just passed one on our.
But yeah. You don't want to try to get going.
You want to break it up, but you don't want to like sit free like that.
No, it's not broken off.
So
So
that we know it hasn't just fallen.
we're just discussing how to get a rock sample. So if you saw us trying yesterday, the rocks here, it's one complete mass here, and it's really hard to break off a piece. But that's also why it's so important to understand what it's made of. So as we do our transect, we're looking for a potential spot to break off a piece of rock.
Can you get underneath there?
And yeah, 400 meters is the transition between the sub-Antarctic mode water and subtropical mode water, which means there are a lot of abiotic differences. So we'll see a general shift at about this depth. So, give or take some meters, we'll see a completely different environment as we continue upwards.
Yeah, so if it doesn't come, that's all right. It's really testing how hard it is as well.
Okay, we're just gonna settle down and see if we can break off a piece of this rock.
Yeah, let's just keep an eye on
Oh, okay.
We'll just keep an eye out there. Yeah, we'll find something really good.
Yeah.
Sort of a
The best time.
crop.
Go off a bit now.
How big of a piece are you looking for?
Just the size.
Most we can, eh?
Just the
Yeah,
fist size,
just
please, not a
huge.
huge shoulder.
Just a hand, fist size.
Seems like a little.
Yeah. Don't know.
This is the what you anticipate anyone else soon. It's called Rob might want it. Let's do this thing on.
Give this a shot.
Yeah.
What do you think about here?
Something breakable in there where the worms are.
During this trip, we have not seen any wrecks or vessels underwater. But you never know. There are plenty of mysteries under the sea waiting to be found. Just gotta get lucky.
Come over some. Have your angle out. You okay? When you zoom in on the camera,
Yep.
keep flying. I can't zoom in. They're getting right between the lasers. Maybe we'll break that off.
Try, but that's pretty.
It's better from this side.
Oh.
Break them up a bit.
Yeah, maybe.
Maybe if you can scrape her face off and then just zoom in and we can just grab a screenshot of it.
Okay, I can zoom in on. Yep.
I can kind of try to break a couple little pieces off and get hit it with the ultrasound. I'm going to try that.
Really, yeah, the number one test is, is it crystalline rock or is it carbonaceous?
How's that? You can see little anemones on the underside and worm runs.
Okay, well done.
Thank you.
Thanks.
All right.
Yeah, if we see something that looks easier to break off, though.
Yeah.
So that was a virtual rock sample collection. Yeah, so under that rock I saw a little anemone, and there were little worm tunnels.
I'll go over and look at that reef.
Are we going up? Yep.
Yeah, we're going up. This bridge is something different than the rest of the surrounding area. So look at that.
Yeah, the bridges are really interesting.
You know me.
Yep.
Yeah.
Easy. Like something.
For me specifically, it's what the top type.
Don't you? Oh, here you go.
That's okay. We'll try to focus on the shallower. Yeah, excellent. It looks like a dead plate hard coral. Probably fell from shallower.
No, that's fine. I really enjoyed it. That's nice.
So fairly soon, we'll see that shift in composition and habitat.
Do you guys want to try that rock that we just went over?
Yeah, if you guys think
possible.
That's a good one.
Rock in front of them. Looks like it could have fallen though. If this doesn't work, there's some problems.
Well, if it looks like it could have fallen, then no.
Up there. That's not how this is right here.
Oh yeah. Okay, we're gonna try to grab this rock.
I'll let you get your arm up there and you can adjust. Go a little closer.
Oh yeah. Looks like you got a good grip on it.
Pretty hard, huh?
Pretty hard. No, it's not worth the arm.
It's a little closer actually. Hold on a sec.
That's right. The rock wins again.
I tried. I don't think we grab one there, but yeah, that's not
Well, yeah. I was like that? Not really, but you know, I'll give you an A for effort.
B+.
Tough grader. I'll scan over this, see if anything's promising on this rock formation.
A little hidey hole. See a fish in there.
Whoa. Got steeper.
That's a good sign.
Yeah.
So the slope is increasing now as we get towards the top 350 meters of the seamount.
see the yellow power ones? You want that? YouTube student thinks it's excellent.
You're right. You're right.
Yeah.
Which screen would you like it? The little middle left.
That one?
Middle one left. Or
Yep. Okay.
that we're also ready.
Hey Pamela, that's a good question. What is this seamount made of? That's actually one of the reasons why we're exploring. We don't yet know. There are indications that it's limestone. It's really, really hard, which is why we're trying to get a rock sample. But yeah, the top and the rest of the seamount is one important question that we'd like to answer. You know, what does it consist of? And more importantly, how was it made? So these are the kinds of questions that people on the ship are interested in answering.
Over here, on track. Little hippie deals over here. Something fish swimming around.
Get back on.
Thanks. Okay, there's a fish.
Want to try this, or do you guys care?
Sure. Yes,
All right.
please. Yep.
Yeah.
Sorry, I didn't realise you were talking.
Right in the middle. Wherever that fish just went by.
Yeah.
All right.
So you might have noticed that the sea urchins, the field of sea urchins has more or less gone away. We're now at 333 meters. It's 16 degrees Celsius. So this is the first indication of some sort of a change in the assemblages as we go shallower. And you could see in this hole lots of dead coral, branching coral that have come from the shallows. So they die, they break off, they roll, and they fall into this hole. And you could also see worm castings, worm runs, worm tunnels on the underside of this, inside this hole.
Here you go.
So if we could break off a piece of this rock. Oh, we did it.
Great.
Fantastic.
Right there.
Both
of these pieces.
Yeah, both please in box 1A.
Sure. All right. Porch out.
So we finally did it. We finally got a rock. So Pamela, we'll get back to you on what it's made of.
All right. That's not good, dude. I think we got a backseat one.
And while we're here, we'll just zoom in on the hole and maybe scoop up some of the corals there, and we could throw it in the same container as B.
Thank you very much.
And you want to scoop with the scoop?
Can you scoop the whole, all those dead corals in there? Just get a big scooper.
Do you want to use the one of the two, or do you want to use the box of the bag?
Thanks, Rob, for mentioning that. It's a good idea.
Let's use the scooper.
And B2?
No, yeah, like,
Or
yeah,
the bag?
yeah.
Yeah. Okay.
Yeah. I'll still this one.
All right. B2.
So yeah.
Just keep it on. Make sure it's
Bridge ROV.
ROV bridge.
It'll work. Just to let you know, we're going to be at this spot for a second, so you can come down to that waypoint, just don't creep any past it. You'll get to that spot.
Alright, coming to a stop on the way. Thank you.
Chris, I think you could buy this robot on eBay. Let me know. Get back to me.
Is it making off?
So I guess whichever is more accessible.
That break was that
Yeah, I was watching.
Oh, you saw it? Scott's on the Slack channel. Nice work. The bosses, I'm sure, are very happy with that piece of rock. Thank you very much.
Do you want me to shut the things that don't have it?
Yeah.
Alright, so what I'd like.
Yeah. Do you rack it on sense?
Not completely, no, I've racked out to get better food now.
Unfortunately, after talking to the ROV pilots, this ROV was built by them, so there's only one of a kind. So you may not find it on eBay.
If they're interested in an ROV, let us know what their application would be to give some ideas. Fine.
Do you want to talk to them about the ROV stuff?
Let's ask specific questions, yeah.
Okay.
But yeah, it is. Sorry. Yeah, we between all the ROV guys, we've worked on many different types of ROVs, whether they be small inspection class or eyeballs, if you will, to full-on work class trenchers, the really big ones.
Is this one of the big ones?
No, this would be a medium-sized work ROV, with more of a delicate science bias.
Right.
The main work class ROVs you're going to have is shielding HDs and UHDs, the Perry Swing B, XL, XLS, XLX. Those are your larger work class ones. And also some ones for specific companies, like Hercules and those specific for random companies. They have all electric vehicles, like East Atlantic, Mohican, Comanche, Mohawk. You can get those various sizes. Those are more of your inspection class. You can get some light workloads like Comanche, you can get light work.
Someone was asking what pressure it takes to break off a rock like this.
The jaws, how much?
The arm, that's about 200 and some pounds, I believe, so...
Pounds per square.
Oh, I don't know what that would be.
The pressure in the pounds per square inch, I have to look at see what it is, see what's down to get it.
The 200 pounds of pressure to break off the rock. And you could see how hard it was to break off this rock.
I'm going to try to go above it to be easier.
Yeah, it's just like...
Okay, you can pull that.
Yeah. Expect more fishes shortly. Once we're done with this scooping sample, we're going to beeline to the top portion of the seamount. Should be really exciting.
Okay.
Like that's scooped up things.
You want to push out a little bit more, you get a better angle? Or
Yeah, maybe just a little bit.
Alright, push down this touch. Alright, move your forearm a little bit.
Actually, that's pretty good, right?
Yeah, let's go push a little bit stairs.
Okay, we're trying to get another scoop here. Oh yeah. You need to scoop in a broom to get it all in. Yeah, that'll do. Yeah, that's good enough.
Okay, let's do 1B, is that alright?
Sure.
1B, or do you want it in the same one
It
as
can
rock?
go in the same one as the rock,
Okay.
because they definitely look different.
Okay, so 1A.
Yeah, so the reason why we're sampling these fossil corals here, these dead corals, is because it's interesting to know whether it's a shallow water coral, which would mean it came from the top, or if it was growing at this depth, you know, underneath the overhang. So when we look at it, we'll be able to tell if it was a deep water coral, and if so, well, then we can identify the species and know that it was occurring in this location at this depth. Yeah, it's a delicate procedure, but of course, once again,
Thank you.
done with ease from our ROV pilots. And you can see a little fish in that hole over there. We're gonna say goodbye to that fish. Yep. Yeah.
I'm gonna say hi. RB2 coming open. Okay. So you go.
No worries, Rob.
Third course. Down a little bit so you can get it. Try pushing in or make sure.
Yes, see what happens with the clothes on top of it. I can kind of give it a little nudge, just a little lift it.
Let me just try to stay by. There you go.
Nice.
Alright, pushing in.
So we're still in the middle of our transect, so we're going to go back to where we stopped before we collected this rock, and we'll continue our transect up to the top. And at that point, we'll go ahead and sample some of the shallow water diversity. So we want to get a good representation of all the different corals and sponges that are found in these different dives that we're going to be doing over the next 3 weeks. So we'll try to sample from black corals, hard corals, soft corals, and sponges. And trying to maximize our groups that we collect to allow us to more quickly document the total biodiversity in the area relative to what our target collections are.
I'll back up a little bit so we can get coverage, make sure we didn't miss anything on what we were doing.
Thanks. So we're going back to our transect point to continue.
Alright, so we're
Hey Eric, Adam here. We're going to continue on with that transect.
Adam, Eric, posing here. Thank you.
Sorry, we're continuing on.
Alright, let's make it.
Thanks.
No worries. Oh wait, exactly the wrong way around.
That's okay, we'll get there.
you're studying with your video.
So at about 360 meters, we stop seeing those sea urchins, and from 360 to about 330, which is where we are now, we're seeing this bare, sloping limestone floor, but we are seeing more fish now. And if there's any fish people on the lines, feel free to shout out the name. All right, and as I said, there were no more urchins. Here's some more urchins.
There are no more really big sharks.
Yeah, we expect there to be sharks at the top of this seamount. It's a pencil sea urchin. So yeah, keep your eyes out for a big shark.
I'm gonna fly over to do some interesting geology over here and a large fish. Come on, large fish, don't go away.
All right, there's another fish.
More fish. I think that there's more hiding places over here, so the fish are a bit plentiful.
Yeah, more fish. We're gonna quickly pause and just see what this. Yeah. So no idea what it is. So we're just gonna take a close look. Let me know when I can zoom in. Okay. Maybe it's soft coral. It's hard coral.
You want it?
No, it's okay. All right. So now we know it's hard coral. Okay. Oh, sorry, people on YouTube and Facebook I think there's an issue with the internet. So we'll get that back up and running as fast as we can.
Both YouTube and Facebook are out right now.
Sorry about that, guys. I think it's back up and running now.
That'll be more interesting. So, yeah, so that's actually someone's trying.
Yeah.
All right. Just promise me the sharks up there not as fast anymore. Okay, I'm going. So going. We have the
And he's pulling up the wood.
we have the pads on the we have the pads on the thanks to the fight with the else.
So you can see naturally made canyons as we go up.
I think Oceanic White, which are the ones that are more
Yeah, I was gonna say, I wonder when that'll kick in. I thought it was about 200. But
So we're at 226 meters. It's 20.3 degrees Celsius, so now it's getting a bit warm. And look at this. So a whip coral, either black or soft. I would guess black by the way it's hanging, I guess you could say. That's interesting. Some more whip coral. The simple answer to what makes these canyons is erosion. Oh, look at that. Fishing net. That's a whole ball of net of a fishing line. So now you could see coralline encrusting algae. So we're now entering this photic zone, this zone where sunlight can penetrate, and that means a distinct change in the assemblages of fish and coral. So we expect to very quickly change the scenery here.
If you're able to, we could maybe slide you back a little faster. We're gonna try to make a beeline for the top here.
Alright, I'm gonna increase to 0.05.
Okay, and then we'll slow you down when you get close to us.
Awesome.
Cheers, thanks.
Alright, so I'm gonna have to quit going up as fast because that's been, but we're pretty good.
Yeah, righto.
Yeah.
So now once we start seeing those corals of interest, we can start grabbing them, starting down, going up. Actually, could we, since we're waiting here, I'm just going to focus in on this branching coral. We're moving a bit. That's all right. Okay, looks like a Gorgonian.
Are you looking to sample this, Jimmy?
Should we turn the water on?
Yeah, just okay for a minute?
Yeah, I'm fine. Those are just white things.
Yeah, we'll keep going upwards if possible.
Yeah, he's water on the back. Someone is just increase the second. Do that.
Okay.
The fishing line will club me as well.
Oh, we got some big fish. What are those, Trevellies? Maybe? Oh no. I imagine this is what the fishers are aiming for when they're fishing on this seamount.
Is that line going that direction or to the left?
Yeah. Yeah.
Different fish right there.
Yeah, lots of fish. So now we're starting to see more corals and fish. So we're looking for specific species because we have a limited amount of time left on this dive. So we're going to shoot for certain hard corals and some black corals that we saw yesterday that are of great interest to sample.
For you. Not a problem.
Yeah, you got us?
Yeah.
So we see soft corals here.
keep it on. I'll put the visual on if we need it.
Okay, thanks.
Coralline encrusting
Just let me know if
algae.
you're about to pay out, I guess.
Yeah, I'm not going to be
It's
paying on it. But I will definitely let you know better view.
Here.
Okay. Yeah. Right.
That's not a bad rock sample if you want one, Jeremy. I could probably snap that off. No? Just say,
We'll look out for one at around 130 metres, please.
Roger. All right.
so yeah, at this point we're going to have to start flying just kind of nice and smooth.
Yeah.
Yeah.
Maximize our time at the top.
We still have, I mean, it's only one, so we still have 2 hours on the top. Basically.
Sweet.
Okay, great.
That's great.
Yeah,
Okay.
65 now. Ooh, big rapid limit.
That's great. Good question. What is the red? So we're starting to see coralline encrusting algae. So now that sunlight can penetrate at these depths, you get animals that use sunlight for growth and for reproduction, which is why you see this big composition shift once sunlight can penetrate the depth at which these animals are occurring. So can we, at the top of this rock face right here, just zoom in on, I'll just look at some of these corals.
Yeah, which ones are you going to look at? One's on.
Yeah, I'll just zoom quickly. Okay, we got soft corals.
Okay, come around to the right. Get you a view from the angle here.
Could we sample that whip coral?
That one right there?
Yeah.
Yeah.
So this is a black coral. It's different than the black coral, the whip black coral we collected a few days ago. So this is a different genus. If I'm seeing it correctly, the polyps are going around the stem rather than just having one row of polyps, which would make this genus Cirropathes instead of Stichopathes. So we're going to take a sample of this colony, and then we'll continue moving upward. And the reason why we're going to collect the full colony and not just the tip of it is because, especially with these few featured corals like a whip black coral, there isn't much information to go on to identify it and separate it from other species. But there's one feature, they're called skeletal spines. So these are spines that are really small, less than a millimeter in height, and they are found on the stem, and if there are branches, found on the branches as well. So we need to see how those spines vary from the top to the bottom.
I'm going with the science campsite. I'm just going to sit down and. Alright.
So now you can see a lot of branching corals here. So you could see they're kind of on the inside of these rock crevices, on the underside, or off to an angle, protected from the currents, the oceanic currents, and that allows them to have a larger surface area without getting ripped apart by the moving water. So that's why you see a lot of these branching corals in caves, in underhangs. If it's
I was trying to freeze right there.
Just zoom them out, people when they get them.
The camera all the way up, make sure we
Look
get a
down
good
there,
angle for what you're trying to grab.
Yeah.
You're trying to twist to the right.
Oh, I didn't realize we were just here. Is it down more?
To the right. Alright.
Was it the black whip?
Yeah, but it wasn't any of these though.
That one right there?
Yep,
Yeah,
that's
you
it.
can point to it with the stick there.
Right, yeah.
There's a little too high. It's right there in front of you.
Yeah. I think we sat on it.
Oh, we didn't sit on it. It was in front of it. I could see it in my stick camera. We just couldn't see it in the size camera.
Ah, sweet. Okay. So it should be easy to pluck it from the base. You know, from the bottom.
Hold on, I'll show you in a second.
The coral base. You need me to point?
Yeah, I can't really come off of the stick.
Point here.
No, on the right. To the right. Middle, right.
I can only see it here.
Yeah, right there.
Yeah.
You see the black base right there? Alright, yeah, I'm fine. I just can't freeze. Are you going to zoom in on it from here?
Yeah. If you just do a little zoom to the jar there.
I can't zoom out anymore.
Zoom in.
On the coral?
Yeah, on the jaw, right? Yeah, right at the base.
Yeah, you got it.
Yep.
Beautiful. And it should fit.
Right there. I'm going to try to freeze where I'm at. See what it does. Alright, we're good. Alright, so I'm going to bring the camera in.
You can go for the big box.
I think it's everything in the big box, yeah.
Yeah, I think we're going
I might
that.
force out a little bit of that. We'll see.
Cool.
Thanks.
Dream work.
Yeah, man. We got it.
Alright, I'm going to push out some more. You can do it.
I think it'd be alright.
Alright. Anything else while we're...
Nah, we'll move up a little bit more. I don't see anything else particular.
It's all looking in. That's weird.
Porches and over there.
Oh, that's a good one. Can't come out.
It's a good fit.
That'd be nicely in there.
Well, after yesterday's coral, we could do anything.
Alright, I'm circling it down. There you go. Good job.
So, the reason why we collect, so we're going to head a little bit shallower until we see the next target that we'll collect. But I'll just explain why we have to collect the whole colony, especially for these whip black corals. So in addition to observing the skeletal spines, those small features that are less than a millimeter in height that vary from the top to the bottom of the coral, and that variation is indicative of different species, we also want to look at the very near the base. There are features called striatum, and those are little marks right above the base. Some species have them, some species don't. So it'll be interesting to see, whether they do or don't have them. And we aren't yet sure if it's even an important feature in identifying and separating species, but that's why we have to collect it now. So, for example, if we're able to put together all the corals that have these features near the base, and then we look at the molecular relationships between these species in a phylogenetic tree. For example, if they all are closely related and they all have that specific feature, that means that feature is informative in separating species between or within genera. So it isn't yet known, but now that we have the molecular techniques to really investigate these questions, we could do it. So it's a great opportunity to be able to look at the bases of these corals. And without that information, we'll never get to understand. We'll never be able to know whether that feature is informative or not.
My son having to fly back because teacher tried to trick me into going to the cave, but we got you. So
Yeah. A lot of stuff in that cave.
we can fly in there as long as I know it's there. Let's fly back out.
Now you got fishing line.
Fishing line to the left, yeah, right above me. Alright, I'm gonna fly off. Bring this fishing line.
Yeah.
Nothing ruins an ROV dive like a fishing line in the first.
Imagine.
Where were we whenever we had that cave
LA.
right above you? You're like, alright, stop.
The best part was
You and Lee were talking about it.
telling the story.
Yeah, our old mentees was telling a story about flying with this huge cable or whatever.
As he was flying in a canyon, which was what we were doing at that exact moment as he was telling.
Yeah. Like, okay. Then all of a sudden, we see this huge cable that's just right across. Oh, that's how to stop.
I imagine getting that in the propellers is bad.
Yeah.
It's crazy because it's ambient light, so it's even earlier.
Turn off the light so we can get the view just for a second.
So I think at this stage we'll try to get to 100 meters.
So we're coming up on the 130 mark, so if we see something that looks like a good there, it's a nice picture.
So that's natural light.
That's it.
So this is the view from the ROV with the lights off. So you can see the sunlight's penetrating at this depth. So corals that need a little bit of light, maybe corals that want to minimize competition with other corals but still need light, will grow at this depth.
Yes.
Anywhere for a rock sample.
Anywhere for a rock? No problem. Alright. So,
Maybe on.
what are you thinking for a rock sample?
We'll find something here. Let's find a good place to land.
Yeah.
I don't think that's gonna
That's not a
happen,
good
but
place.
I think we're just gonna have
Well,
to.
you just crest the top of it and we'll see if there's something up there. I just didn't get it because no, I could get it.
Yeah, it's amazing how many overhangs there are in this part of the seamount. You know, when you think of a seamount, especially one like this that's so circular and has a point at the top, you know, you think of like a Hershey Kiss, but this is complex, which allows for more, for higher biodiversity.
Knob. I don't know if there's anything.
I haven't seen anything that's, just keep coming up when we don't mind there.
Yeah.
Okay, we got some trevallies. I think the trevally is tagging along with this alien robot.
Yeah. We're gonna plug it and get once over. Alright, come on. Right. Yeah, I mean just fly right. Look at this nice coral for you though.
Yeah, I'm looking at that as well. Maybe you can find a rock in that area.
Maybe.
Oh yes, Caragoides ferrado, of course.
Brilliant, ROV. Yeah, just, I think what we'll do is probably get you to just hold there while we crest over the top here.
So,
just to keep us stretched out.
not my problem.
Alright, sounds good.
That fish that line right there.
Okay, yeah.
That's a ship flare.
Then we can keep going up.
I will do that for
For
you.
the next one.
Whatever other color.
What color would you like?
No. No.
Just so it's visible.
Sure, I'll see what I can do.
Alright, thanks.
Thanks.
Yeah, that would be a bad thing right there.
What's that?
That line.
Yeah, we gotta be careful. Just fishing lines don't work well with resters.
Absolutely.
So,
Copy that, Ben. They stretched out there?
Roger that. Looks good. You're still okay.
Yeah, I think the rope is right there. Okay, so we're going to continue on. Yeah. Should we go left or right, you think?
If you go straight onto the ship, that's probably the best.
Alright, so continue on to the right then.
And you're looking pretty much in line with the ship. You get right. Is there that line there?
Yeah, the line. I just passed it. So you just do off a little bit to the left of the screen now. Alright, I think I started moving up a little bit. If you see anything that you think you can pull off rock-wise, so I can nudge up. Turn some good lighting.
Yeah, now we can see. Maybe an edge.
Yeah.
Maybe an edge here.
But I just don't really see a rock. Do you want to collect any of this stuff? I don't see a blue rock option except for maybe underneath there.
I wouldn't waste too much more time. It sort of needed to be around 130, 120 metres. We're above that now, so I just keep going.
I can drop back down if you want. If you want one at 130.
If we can target something around between 130 and 120.
Yeah, then come on down.
The reason for that is it's a past sea level.
I am not going to be able to do this.
You're fine.
And then after we get that rock, when we're coming up, we could probably grab that bushy coral there.
Is that a scientific term, bushy coral?
Yeah, scientifically called bushy
Think you
coral.
can pull some of that right there?
Yeah.
How about that guy right there?
Yeah.
All floats emerge again.
Not that, Ben. We're just diving down to get a rock sample here.
And we want to keep you engaged.
Yeah, we're not going to pay out because you got mutual engaged, eh?
Roger that.
Okay. If we have to pay out, I'll let you know first.
Roger. Thanks.
So there's a lot of corals here as we're searching for a rock to sample. I see a hard coral here. Covered in sand. I'll zoom out so they can see what they're doing. Lots of gorgonians. Sponges.
Yeah, sure.
Alright, so rock two.
I don't see any cats. At least not any handsome cats.
Underneath.
Yeah, if you want to grab the coral with a rock. 2 birds, 1 stone. Pretty close. It's
Do you
good.
want that coral as well, Jeremy?
You might as well.
Grab some rock with the coral on it.
Yeah.
Yep.
So we're going to go for 2 birds with 1 stone here. We're going to get a rock sample that just happens to have a coral.
Let's find a better spot. Let's just leave it.
And you could
What I can do.
so inside this hole you could see some branching gorgonians and branching black corals. So as I said earlier, these branching black corals and these branching corals in general love to be in areas that are protected from currents. There's also very little known about that because they're really hard to sample. I mean, you can see now with the ROV, it's very challenging to work right near the wall and especially underneath the wall. And when scientists go scuba diving, a lot of them overlook these hiding places. So, they may search along the top, but not have the forethought to look underneath. So, less is known about corals that are a bit more cryptic.
Sorry, that's awesome. I'm so glad that you put that on there. That's my new favorite thing. Alright, I have no guarantee it's gonna add whenever you start yanking out a rock.
So you're going for that rock?
There you go. And then we can get whatever.
Just get the rock
If you
with.
sit cam down, will I be able to see it?
Cam down? Yeah.
Maybe
Try.
not, it's too on the left.
Try. Hold on.
So we're
Chin and then ram in if it would make it easier for you as well.
so we're at 120 meters, and it's almost 25 degrees Celsius. Pretty warm. Get up there.
Oh, that's slick.
Like that. So, sorry, we're just very happy with ourselves at the moment. I had to put on these new bumpers on our Rams that normally carry, and so now I can use that as a fist
Right.
to pull myself in place.
Awesome.
So he does that.
Oh, there that goes.
I can go get it. I see it. I can
You see it? Yeah.
I can see it.
Sure.
It's attached to the core.
Yeah. So, all right.
Yeah, you can hold it up by the coral.
Sure. All right. Flying off.
Do you want to center up the camera, just give him a little bit look down if you wouldn't mind, Chris?
You got it.
Zoom up, yeah. Look around, yeah.
Yeah. There's a bunch of fish there just waiting for us to keep breaking up.
And if you just come to the right there, Jeremy, that'd be great.
Perfect.
And you see the coral that's standing straight up? I think that's the rock, so I might just grab the whole thing if that's cool.
Yeah, go for
That's
it.
cool. Thanks.
Just pull your veil up there.
All right. I don't know how we're gonna reach this.
Just separate these here.
The problem is this. Right there. So, hold on. I can try to come over here. Can you reach it? I got a slab around there. Yeah. It's not gonna help us anyway, but let me just try to get up in there.
So the rock fell down with the coral attached. But it's really close to the wall and to this crevice here. So we're gonna try to sneak our arm in and grab it.
I'm on. Yeah. I can't do anything else with my hands though, so okay, come on.
Or just look a little bit right there for me, Jeremy.
Right?
Yeah, thank you.
And you guys have got good eyes.
I'm gonna zoom in.
Yeah, great.
Alright, I'm touching the top, so.
All right.
Got it?
Yes.
Great.
Look at that.
Alright, so I'm gonna.
That's superb.
Oh, and there's
Something for everyone.
and there's little hard corals. So these are A's and Elliot cup coral. I guess solitary hard corals, which is amazing. And there's a lot of living stuff on this rock. So this rock will keep us busy for the afternoon.
Actually, if you first, I'll just. Alright, ready? Yeah.
Yep. So we're collecting this rock sample at about 120 meters, which is the last glacial max sea level. So it's a really good depth to sample. That's all right.
We open up 4 and then
slide it in there.
Alright.
Because if this was what sea level used to be like, we can compare rocks at what is now sea level and what used to be at sea level to understand how that change occurred. Are you guys stable right now?
Relatively.
Can you grab that? This? Just a branch. Just take like a fistful here. If it's too hard, it's okay.
That might be. Yeah, we can do it. We just got to reposition. I think.
That's okay.
I think it's back in that hole, but let's have a look.
I can come in with the torch, see if you can get in there. Just gonna look up to make sure I'm not. There's this right there. Don't worry about the portion.
So while we're here, before we move up to the shallows, I see this is the first specimen representing the genus Myropathes, family Myropathidae. So this is the first Myropathidae specimen as well. We're just looking up for a second.
Is that black coral, Jeremy?
It is a black coral.
Myropathidae?
Yeah, Myropathidae. And so this is a shallow water species, but this is probably the deepest this species can occur. I mean, you know, who knows how deep specifically a coral can occur, but this is deeper than it's normally seen. So we're going to try to sample it.
I'll try it with the starboard arm, but maybe not somewhere. I may not be able to get it with the starboard arm because that's where our snippers are.
Oh, if you could sample it near the base.
I may not be able to because that's not on this side.
All right.
But I can try to fly off if you need me to.
No, if it... Beautiful. All right. So yeah, if you can grab by the rock, that'd be fine also. So we've collected species in this genus in the GBR, but this is probably the first time it's been collected from the Coral Sea. So that's interesting. In the back. Yeah, in the back is fine. So what we can do is compare this specimen to ones collected on the Great Barrier Reef, and we'll see if they're closely related species or not. So one interesting question is whether species are more closely related based on close geographic extents or bathymetric extents. In other words, if two species are at the same depth, are they more likely to be closely related than if they're close geographically? It's an interesting question. It can explain how species ranges, how species can connect between one ocean and another, or from the deep to the shallow. So that kind of stuff is just starting to be studied. And so, looking at coral specimens like this that are found along relatively large geographic and bathymetric ranges can help us answer that question.
Pull off and then. Alright. Alright, I'm gonna open back up and let you put it back in.
Yeah, you want to land out somewhere?
I can try. Yeah. Yeah, we're not gonna do anything else at this depth, right?
No.
Alright, I want to take them off.
All right, we've collected soft coral, black coral, and rock at 120 meters. So we're now going to head to the top.
What's that? Alright, there you go. Yep. Down, sit. Alright, there you go. If you open it some more, you get. Yeah, maybe. Alright. Watch out, watch out, watch out, watch out. There you go.
There's probably a branch stuck in one of the.
I would go left. Out of here on the load on the side of the arm. So, what you could do, alright, stay right there, stay right there, stay right there.
Yeah. There you go. Now that is that's how you do it.
I'll get back in there. It's alright,
Yeah, all right.
Alright.
Onwards and upwards.
Alright, so we got the rock at the 120 to 130.
Yep.
Full corals.
So the last stop on our tour today on Cairns Seamount will be the top.
Good job. What I'll do is I'll just fly with my stick camera, make sure you're not going to go into a cave, and then you can look around wherever you want.
Sure.
And
Fish.
we're not in a horrible spot temperature-wise right now, so
Okay. We definitely want to go up.
yeah, it's not what we should. It's not what
We're good. Yeah. Okay, cool. That's good.
Control deck.
Yeah, go ahead.
Hey Ben, I'm just gonna start picking up a little bit, so keep an eye out for those folks.
Roger that.
Thank you.
Broke fish. Out of the tanks. Backing out slowly.
Okay, we are now ascending. But the dive is not over
Copy
yet.
that, Ben. Are they in a good position?
Yes. Stretch out. Half straight.
Yeah.
Yeah, a lot of fish on this area.
So
Started to scoot over to the left and
yeah.
Nice little hang right there.
So once we start seeing some of these plating hard corals. That's one of the targets for this dive. We're looking for 2 different genera. We're going to look for Leptoceres and Acropora. And now we're starting to see some of these coral, these hard corals, which is a good sign. Okay, so we're starting to see this Leptoceres, a brown circular coral.
50 yards.
So,
So I'm gonna keep him where he's at.
So I'm getting
So we're starting-
messages from onshore to ask for another rock at around this level if we can. Any
So-
of those sticky outy ledges.
if we can get a rock and also one of these corals, that'd be... Yeah. All right, so we have found our target, hard coral.
What if that told you that's 56?
Right now. So you're gonna go with your tower report on.
So just point again at where you're looking to get, Jeremy?
Yeah.
That's brown coral.
Brown, okay. And they pretty brittle?
Yeah, if you flick it, it'll flake off. And then maybe, I don't know the best way to...
Let's go start with that. We don't get back.
That's fine.
Yeah.
So,
Try and get the rockets.
Yeah, exactly.
Maybe hard on this,
There's-
but we'll see what we do.
Yeah.
there is a little bit of an overhang right where I'm about to try to set up that looks like you can probably pull off the rock that we
Right. Yep.
Yeah, and any of these corals will be fine. How many expeditions have I been on? During my PhD, I've been lucky. I've been on a few expeditions. I was on the ROV Investigator this time last year, also in the Coral Sea with Rob Beeman. I was on ROV Elise out in Lansdowne Bank, which is west of New Caledonia. And that was deep sea expedition as well, which was really amazing. I've also done some scuba surveys on the Great Barrier Reef. So with Tom Bridge, who's one of the principal investigators on this cruise, working on that project as well. So we surveyed the entire Great Barrier Reef on a little scuba boat, or the smallest research vessel you can imagine. And we surveyed corals from the bottom to the top of the Great Barrier Reef.
Yeah.
I have not ever been in a sub, but that's next on my wish list after this. Being on Falkor has always been something I've wanted to do. It's an amazing opportunity to collect the corals that I'm working on in pristine condition.
So what do you want to collect, Jeremy?
He doesn't want to collect
Just the rock.
It's not.
You're calling Bridge, eh?
Yes. Hey, Anna, maybe you could ask the, Joe was asking if you could send in a deck officer.
Onto that deck? Yeah, sure.
You're about 25 meters away.
Okay, Bridge, ROV.
Right, is there...
Did you copy that deck
Do you guys want?
from the deck there?
What are you targeting for
Say
the
again.
coral?
No, nothing
deck
here.
is
Just go for
looking
the
a
rock.
deck officer
You're not targeting
very-
anything here.
Very close to the floats on the
So
back
you don't
deck.
care if it's
All right,
a
so
coral or not?
recovery.
Sort
Not-
of, yeah.
not for a while, but-
Half recovery. Just
Anything that you
recovery?
can break off.
Okay.
Anything
Yeah.
that's- anything
Two
you
take
can
the
get
phone
in
and
some
put it down.
samples. Anything from what we store in, if it happens to have coral in it, that's fine.
Yeah, totally.
Would that there be...
Yes. You mean that?
Well, no.
Yeah.
Or you want the lower stuff?
Oh, it doesn't matter. Whatever you, anything here that you can
Yeah.
use.
Yeah, so you can do the port arm, so if you're the easiest thing from that.
Where do you want?
Any rock that we can do like a hang overhang if we get the port arm better, if not rock.
So yeah, for us, we're really interested in what the rock is underneath the coral. Coral and biologists too.
Yeah, exploring as a part of my PhD, our PhDs and careers, when you try to answer fundamental questions, you know, like what is something made of, or how did something originate, you get the opportunity to go exploring. You know, we know a lot about certain things, but we also know very little. So if exploration is in your blood, there are plenty of basic, fundamental questions that anyone can answer.
Yeah, we'll find a better spot. Because if I even torque the vehicle a little bit, all that leverage is on that road.
Yeah,
It has to be in this depth at 100 exactly.
Around within
One of those other teams,
100,
right?
within 10 meters of where we are right now.
Okay. Let's see if we can just find something a little bit more opportunistic. That was just real hard because we're just kind of forged on this one little accident as a lever. It's not a great thing. We can find a real good place to perch.
Yeah,
As soon as we grab on,
You're doing that shit. Yeah.
Chris tends to hit me when I knock him off the wall.
Well, I lined up right here, yeah. Then we try to go above us. What do you think?
Because we're so shallow, it's going to heat up a little. Let's try to find the most opportunities.
I think I like that. There, great.
Just keep sliding to your board there.
Board?
Yeah. Push it line up to ship. Yeah, taking it down.
Board or my starboard?
Your board.
Not a lot of opportunity.
Just because we've had experience on how hard it is, you've really got to find something that's snappable, you know?
Yeah.
We'll flap over this little ridge here and see what's in that. Hold on, I'll get by this rope first. Yeah, I'll get by this rope.
Oh, yeah. Okay, yeah.
We're on the Cairns Seamount. Cairns. Yeah, us Americans would call it Cairns, but apparently Australians like to drop the R when there's an R in it, and when there's no R, they like to use the R. Very complicated.
Accurate. Alright, what's your starboard? No, your starboard, moving out of the way.
Look, Vanessa. So we're at 100 meters. It's almost 25 degrees Celsius, and we're nearing the end of our dive, but we still have a few targets that we want to hit. So we're trying to collect some hard corals from the shallows. Our time is limited when the water is so warm, so we have to be really careful about how we use our energy and, avoid heating up too quick. So we're just looking for a rock sample here.
Good, you got us?
Yeah, how much longer until we start recovering? We're looking at.
Don't know, what were they saying, off the bottom of 3 or something? What's the very core? I don't know, check with J-Rod.
What time do you want to come off bottom?
As late as possible.
It's up to the scientists to get the AUV in, so.
Oh, okay.
Yeah.
And we can't come up very much more, so.
Okay, yeah, we're getting lots of samples here, so we'll see what they want to do from there.
Okay.
Our motor temp is actually fairly good, but like you said, we're in 100 meters of water.
You've got floats are almost at the dock and in.
Are they?
We started taking them off. We did that before.
The one thing is we're keeping the ship where he's at, so if we could stretch it out a bit, we might be able to
Kind of grab rock here.
pay it out a bit. We'll see.
Yeah.
Okay, because we have lube down here, we got everybody down here to recover whenever.
Okay, we're just taking a sample real quick.
Okay. We're doing samples.
All right, we're going to try to grab a rock from 100 meters.
Yeah, I see that.
Oh, yeah?
Oh, that's still wet.
Let me know if I can help.
Yeah.
Good piece. Alright, fine, it's a little bit more.
I'll come in.
Whoa. You want that whole thing? I want that whole thing.
Want that, Marty?
Well,
Pick the best piece.
no, sorry, abort.
Alright.
No good?
We want to break a piece off the ledge, please. Is that just pulled out loose?
Pulled it out from underneath the ledge there.
Yeah, it was part of it.
Yeah, I think it came off there.
Jody says no.
Alright.
So we're trying to pick the perfect rock.
I want something solid that's less weathered.
When you touched it, it was already wiggling.
Yeah.
So we don't want that.
It was just scary. That's all.
So it's a bit wobbly.
I don't think you reached that high. That's what we need to
Okay. All right.
The whole thing's rubbly.
I thought you wanted a big rock.
Okay, so no good.
Yeah, it's okay.
Should we?
So, yeah, if we can
Find a different location.
Surely that's in situ.
Well, it could be slumped down the whole thing because it's loose.
Okay, no, thanks.
Okay.
Thanks for
So
trying.
days and they didn't go for it.
Thanks for trying. We'll keep going.
Okay. Just while we're here, do you want to just throw it in the bottom box anyways? We've got lots of room. No? Okay.
Sure.
Jeremy's like, I don't want to trust him.
Well, it may. I mean, if you don't think it's going to be useful,
It's not useful for us.
good
It's
not to
useful
put it on
for
top of
you.
the corals.
Hey, don't call a landslide on me, dude. I'm gonna fly away. We're just gonna
Yeah.
leave that there.
Yeah, no one wants the wiggly ones.
Yeah, it's a hard ask because the nice cliff exposures are so hard. And then these bits are obviously weathered.
Yeah.
So now the rating says yes, take a bit anyway. So if you are able to
There's
go to the next
another
one, piece up.
overhang. There's an overhang right here. So might be able to, yeah, I don't want to go back over to that spot because it's collapsing. Well,
What about this tree right here?
that big rock right there? Two fish underneath it?
Yeah.
Yeah.
I don't know if it's cast or not.
We'll find out.
Yeah, let's take a look.
Maybe further around, you've got a cliff.
Alright, one to the left.
Just, I'm also conscious that we're sucking up biology time as well.
It is what it is.
Alright, right there, some more of that overhanging stuff.
Too much rope.
Oh yeah, you're right there. Yeah. So is there a lot of fishing done over here?
Yeah, there used to be, there still is, there used to be a lot of longline fishing out there. Because amateur fishing is allowed. I think it's no longer allowed to do commercial fishing. Not sure.
Yeah, I haven't seen this much line and rope debris since Louisville. Put it here. Sure. Do a rope here. There's a bunch of rock right here, but I don't know if that's really long. More rope right there, too. Yeah, another piece here. There's one right there in front of me. Two of them, actually.
Yeah, that was like it's all flips.
More rope. This. Jeez.
Yeah.
That'd be our best interest to take a knife down like that.
Yeah, really. So we're seeing lots of fishing line, which is making it really challenging. Ooh. Really challenging to navigate.
Get out of there.
Yeah, this is dangerous.
This is getting risky.
Yeah.
So,
Yeah, totally understand.
okay. So we're gonna just press the top then?
Yeah, okay.
I want to do a bit of a distance.
Yeah, yeah. Ben, you got me?
Roger.
I'm going to start picking up real slow. Let me know. You're going to take some floats off, I suspect, then, okay?
Okay. Is
Do you want to take over on the winch then, Ben?
Roger.
Okay.
So, to avoid
You ready for me to take control?
potential
Yeah, go ahead.
catastrophe,
Roger. How high are you coming up?
we're going to come up to the top.
We're going to try to broach the top here, but we're going to do it at your speed while you're taking the floats off, so we keep our delta kind of 50 or less.
What's your depth?
Depth right now is 93.
Could we go to there? Is that safe
How
or no?
far is that top pinnacle?
No. Yeah, look down.
Oh yeah,
Just,
there's the line.
let's just stay away from all that.
That's fine.
Sorry, what was that, buddy?
What's your depth of that top pinnacle?
I think it's 60 again.
60.
Yeah, it was the same as last night.
They're coming up to check out the top. I don't know if they're recovering yet.
Yeah. That's the last thing.
So that's your umbilical line?
Yeah, that's what it is. Yeah.
Good question.
Yeah. That's That's the only rope.
That's the only good one.
Huh? Yeah, then I'm pulling in.
Yeah, that's we can see that. Yeah, okay.
You're doing great. We got a big belly behind us.
So we're gonna head towards the top and try to sample some of the shallowest corals on this seamount.
Alright, here's a crest
The
right.
other thing is if you stretch out here.
Yeah, so they get over this. You can pull the ship forward a bit too.
I think once you
So we're at
it'll stretch out.
80
Then you take the
meters
float off
and it's 25 degrees Celsius.
Alright,
The ascent is going to be really short. We're not done yet. We still have some sampling to do.
Yeah, Mr. General, are you to go for recovery now, or what are we doing?
Some more samples.
Yeah,
we're going to try to sample on top here, but we're just coming up real shallow, so we're going to take some floats off.
Alright, sounds good. Sure.
So what do you after?
So I think this is a Leptocereus. Yeah, this is a hard coral, a relatively deep scleractinian.
I can look at it, but I can't get it closer to the white rope right in front of us.
Any of these? So this is all Leptocereus, like that.
Right there.
Yeah. Maybe left or right.
Anything close to rope?
I got no
Yeah, it's no go
all the way around it.
Yeah, so
Alright, we're gonna take one of these floats off, maybe two.
I'm gonna stand by on that because I'm gonna go forward.
Yeah,
See
we're
if we can
try to stretch it out a bit.
Well, the floats are at the docking end right now.
pay out a little bit.
Let's grab one up.
Okay.
One up.
Alright, that's fine.
So it seems
I'll stop where I am.
Leptocereus is covering the floor here. So it'll be fairly easy to sample one from here. So we're gonna give that a go when we find a safe entry point.
You reckon we're gonna keep moving, Cody, or still taking it off?
very turbid.
We're gonna take one of these floats off real quick. Get into the water.
No worries, bro.
Yeah, once we just crest over the top, Cody, I think what we may actually be able to do is pay out and just let those sit on the top of the surface, and we'll just go forward, keep the ship where it's at.
It's shallow, huh? No one told me.
You got that, Cody?
Alright, we're taking one float off.
Yep.
Alright, you start airframing it back out. I want to check up the top.
Alright, Cody, am I clear to fly forward?
Yeah, bits are up.
Alright.
Yeah, Aquapora and this Leptocereus, and also cup corals.
Yep.
Alright. Big line right in front of me, so I'm gonna fly up over it.
Yep, so we're right below the top of the seamount. So you can see lots of fish, lots of action, a lot of movement, high diversity of corals. So obviously you can now see this transition into a hard coral dominated habitat, but there's also branching black corals and branching soft corals.
How's that, Chris?
Okay.
It's looking good, man.
Alright, man. I'll stop there.
Yeah, just my season anything. Alright, now once we crest over here, it looks like it clears up, so I might be able to just find a spot that doesn't look dangerous.
Those starting to stretch out, Cody?
Yeah, Cody, how's flows looking?
Are y'all coming up any higher?
We will be a little bit, but not much.
No, we're at 60, so we should be fairly close to that. Should be the top.
That's good sample.
Okay.
Yeah.
Alright, there's if we're coming up any much higher, we're gonna have to start taking on some more floats, so just be worried of that.
Yeah, I think we're at the top, so if anything, we're just going to stretch it out on the surface.
so he wants to collect right here, the woman to the right.
Okay, just make sure there's no real.
Yeah, I'm looking right now.
One over there, but not far away. So.
Yeah, I can sit right. Is it alright if I just sit down on this?
Yeah, I think so. Yeah, just below so we can look high level at that.
Yes. You want to collect, I'm saying, right?
Yeah, correct.
Is there any union?
There's something coming to different, a whole bunch of different coral species. Monticores.
Yeah, we want Monticore, Aquapora, get a little red pin. Awesome.
Booth on his way right now, so we won't have someone to run this A-frame, so what you got with the floats.
Yeah, I think if anything, we'll just stretch it out.
Okay.
How many got on the surface?
So we're just trying to position ourselves to be able to collect these hard corals.
Just let me know if you need me back there. I can be back in a few minutes.
Are you going to use the starboard arm?
Yeah, we're going coral.
Yeah.
You got one relief in there? Adam? Chris?
I'm alright.
Yeah, I'm alright.
Thank you. My perspective is a little bit off down at this level. Alright.
Yeah. So it's obviously gonna break really easily. So I guess try to take like half of it. You'll probably only get a quarter.
Rock?
No, the coral here.
Mushroomy looking thing.
This or this.
Sure.
So we're going after this plate coral here. It's a classic mesophotic coral, hard coral.
Where do you want to put it? 3 and 4 or?
3 and 4 is fine. Yeah. The lasers are on, yeah?
Yeah, that should verify.
I'll zoom out to see it. That should be down, right?
Alright, so.
You looking for what's up?
I'm looking for the.
The laser. You can grab it. It's okay. You can grab the. Oh, there it is. Okay, thanks.
Yes, right there. Zoom in.
Yeah, I gotcha. Okay.
That's stretched out. Deep portion.
My portion.
Yeah.
It will. Yeah. That's kind of important.
Oh yeah, but I think they're traveling into Capstone.
Yeah. So locked. That's too small.
Too small?
Yeah. Yeah. Try to grab.
Try to get
Just across the rock there.
It plates outward. It can be across, but it seems like it's. There's a bit to grab, I think. The texture of the coral. So it has these little coralites, so it's a little rough. So you're gonna have to grab a lot more than you intend to, because you're gonna crush what you touch. But whatever's connected to it that doesn't fall off will be what we look at.
Yeah, I'm gonna have to get closer. I think it's sitting on a rock too, so as soon as I get in there, hit something behind it.
Alright, check. I'll forward a little bit.
Yeah, and I think that comes from the top down.
That's nice.
There.
Yeah, let us know if you need more build.
Okay, thanks, Corey.
We got a pretty decent delta, so I think we're alright.
You can get that.
You want to get that one?
Yeah.
Yeah, that's a better one.
So the coral's really fragile, especially with the claws that we have, which, you know, are great for removing rock, but for thin skeletal structures like hard corals.
Yeah, you don't need to cut with this. You can just try to grab the you know what I'm saying?
I just feel like this one at least is better squishy. You might get a better grip on it. The other one, just crush it and drop
Yeah. As long as you don't, it won't, because it'll still connect through the tissue. So even if you break the skeleton, it should still hold on. So if you just try to grab the meat of it in the middle.
Oh, there's really back down. What the?
the elbow before, just be able to reach.
I can porch out. I'm still pretty far away, actually.
That's gonna be fun.
Right, stand by. Okay. Yeah.
Too far?
Yeah, I didn't realize how far over. Really quick.
Okay. Yeah, this is certainly scuba diving depths. It's a little deep. It's still 60 meters. Not impossible to dive to collect to, but a little bit deep. Cairns Seamount, there are strong currents, so you know it's not the easiest of dives, I assume. It's also very sharky, which isn't a problem either. But yeah, since the ROV is here, we might as well grab a sample. I'm just going to zoom in on the coral of interest.
You got that. I'll link you there. Alright.
Yeah, that looks like a good one. Yeah, it would certainly be a decompression dive, which again also isn't a problem. You could even flick it, just go from under it and flip it up and it'll break right off. I don't know if that'll be, yeah, just straight up. You might be on the under the rock now. I don't know if you want to get a little closer. Yeah, there you go. That's it. Perfect. So we just grab that piece that fell. There it is.
And now I'll keep on grab that start.
Alright.
So let's do.
There you go.
Yeah, we're looking for all the hard coral genera now. So we'll try to shoot for a little of everything. No, yeah, these corals are really fragile. They break really easily.
Yeah, it's 55, so. As soon as I pick off the
Straight back.
Alright, I can porch in a little bit. You ready? Oh, you got it? Just dip down.
Just gonna try to get rid of the out of it.
Get that underneath as well. That rig guy. Alright, good job. 1B. Alright. Porch out just a little bit.
Or sorry, 1D.
Alright, porch out just a touch. How's that for you? Yeah, feel it?
Yeah, it's great. All right. Can you just pan left and waft?
You take that. You take that. Just shot. It's just hit 56. But we got sitting down, collecting. So I think once I come off and move around a little bit, it'll drop a touch.
I think the arms are heated up.
Yeah. Almost,
What do you have on
I have all everything over there on the ground. We're very precariously perched on a little knob.
All right.
All right.
Can we? I guess.
Back off. And
Should we walk that Goniopora? Say again. Should we try to walk the Goniopora?
Yeah, no, it won't pass.
That's it. Oh, it's okay. So let's.
I did get a side view of it, and it's not going. I don't think it is.
So is there
not
like
Nevada
certain things that are like really?
all right.
So.
I think this might be one sample.
Yeah.
Just because we're climbing up and above our thing here, so.
Yeah. Andy, Jeremy, we need to make a decision. What's your highest priority in sample because it's getting too hot.
You reach that. You see those two ones?
This is it for right here. So you can grab that.
Yeah, right
So
there.
that's.
Just any of this stuff. Do you manage to sample any of the series?
Yeah.
So this is local service right here.
All right, I'm gonna try some real fast on.
It's like one Flora.
All right, so we've been trying to get a few different genera of hard corals. So we're looking for Leptoceres, which seems to be this one here, and maybe one of the front ones, yeah. And we also have Pachyceres in the same location that we're going to grab right after this. And yeah, this is what we've been looking for. So this is a great grab.
There it goes. Big shot.
So it's in C. Yep.
All right, you said you wanted to do a little bit that left.
That other piece?
Yeah, the one that how it crumbled.
Yeah.
How you doing those numbers?
57.18.
Right.
All right. Nice work.
Yeah, awesome.
Alright, well, let's try to do this one in C as well.
Yeah.
To differentiate.
Yeah, please. Yeah, these hard corals, yeah, good if we separate them. It's not my specialty, so. Yeah, it's absolutely possible to see a seahorse here. Certain seahorses obviously associate with certain corals or seagrass or algae. So if we were to find the specific sea fan that the pygmy seahorses live in, we could probably find them here. We certainly don't have the time to look for it on this dive, but we've got 3 weeks, so we can look for them in the next upcoming days.
Where's it going?
1C. 4 deep. Just not where we put together with the water. That's perfect right there.
Nice.
Alright, bro.
Can we just zoom out for a second and look at the rest of the area? So if we can maximize.
What I'll do is zoom out and I'm going to fly up a little bit.
Yeah, okay.
Just to move the oil around. You know, It's a little hot.
It's the east, so.
So there's a lot of different species and genera of hard corals. Of course, there's other orders. So it's a really diverse area. It's hard to choose which ones to collect in the little amount of time we have left. So we're going to try our best to just maximize our sampling effort.
Cody?
Yeah, go ahead.
I'm going to fly directly away from you, about the same depth, so it should just stretch us out.
Alright, Lynn, you got that?
Roger that. Let me know if you want something umbilical, Chris.
Roger that, I'll let you know.
What's your tip, Tread?
56.
Okay, so we'll drop a little bit.
Yeah, I just got off the seabed and
Alrighty.
So, yeah, our last target is an Aquapora. So it's another Scleractinian genus, and we're just going to keep our eyes out for them. So, I guess if we can get as close to the floor, the sea floor, as possible.
Maybe closer is going to mess our visibility.
Yeah, right.
So soft to build it. I'm just happening to sing them all.
Yeah, no worries. Yeah, we can keep looking for seahorses. I love seahorses. Yeah, we can find Aquapora at 50 meters. I think we saw it yesterday, actually. Towards the end of the dive, we saw some Aquapora.
Chris, she just want me to give you 10 meters or so, because you're starting to pull it down yet.
That's fine, yeah, take me out a little bit.
Okay.
Looks like we just passed over a branch. Crop around. Underneath. Yeah. Gone too far.
Oh, actually, yeah, maybe. Yeah, let's look in this area.
10 meters.
There's a plate
Oh, that's it. Yeah, that's it.
Is this what you want?
Yep.
Alright, I sit on the other side of the foreground.
So we think we've spotted an Aquapora. So they branch, but they branch in a plate. Often a lot of the hard corals have a plate morphology, and that maximizes the amount of light that they can capture. So they have a larger surface area.
Just have to be careful with that one, Jeremy, because if you break this will take the entire colony, it'll be super.
Yeah, so similar to the other hard coral, if you want to grab it from the middle.
The one next to that big one?
Yeah, this one, yeah.
Nice.
So I need to get quite down to
Thanks.
the big ones.
We'll probably go start around, right?
Yeah. Alright, so the stuff that I want to.
All right. Thank you, Gus Crosby, for finding this Aquapora.
Hey Ben, do you want to give us another 5 meters of umbilical?
How much money do you have?
$1 a meter.
Okay.
Stop, yeah.
But you didn't specify currencies.
That'll just.
Yep.
You said dollar. I agreed to $1.
Not US dollar, it could be.
Australian dollar. I'll take Australian dollar.
It's a really low value dollar right now.
Canadian?
I'm not greedy.
Yeah, you are.
Sometimes. 5 meters.
Thank you.
Roger.
That's good.
Ceratopra hystrix. What looks like Ceratopra hystrix. And possible coral.
Yeah,
What's this?
that's a soft coral.
Soft coral.
We can grab the hystrix after, if we can grab it without moving. Yeah, it'll crumble.
Yeah, possible. Yeah. So, 1 or 2%. Yeah.
So we're just trying to really maximize sample as much as we can because we don't have much time left.
Yeah, you don't grab it, so
Yeah, we're shallow for a long time, and we're definitely nearing our time. So we're gonna try to grab this one and anything else nearby before we have to come up.
They come like this.
As deep in as possible. And lift that way. The lift that way is just going to the whole. It's just going to crumble. It's coming out from, it's growing out from this.
So,
It's
brain things.
I just lift straight up underneath.
yeah, there's far up.
I think you mean the soft coral to the bottom left. Those are soft corals. Yeah.
Really close, and before I get some more.
At this depth, this is a pretty deep depth for hard corals that require sunlight to grow. And yep, so the deeper they go, the more plate-like they will be in order to, again, maximize the ability to acquire sunlight. All right, so this coral is fairly fragile, so we're just trying to figure out the best way to go about collecting it with this robotic arm. If we were scuba diving, we would have a chisel and we would just tap one of the corners and a fragment would just come right off. With a robotic arm, it's a bit more of a challenging procedure, but nonetheless, we'll do it. While we're collecting that hard coral, you could see right below there's that whip coral that looks like it's dead and overgrown by algae. So that used to be a whip black coral, and you can see now it's very hard. And if you were to break off the bottom, you could feel how dense this black coral is, and that's one of the reasons why it's harvested for jewelry purposes. Yep, so except for that soft coral that I pointed out, the rest all seem to be hard corals, different types of hard corals. So I think Unknown Explorer, the one at the bottom left, is that what you mean? Yeah, that would be an encrusting hard coral. Beautiful. If it's easier, you could probably just throw the whole thing in. Yep, so now we're going to see the ability of this arm to balance a crumbling hard coral. That's pretty good. So you could even see, so the corallites, the ends of the branches, you could see that they're almost fused, which is indicative of a specific species. So that's one of the features that they'll look at, you know, how elongate are those little sub-branches at the ends of the branches. And again, coming back to being a plate-like form, just the more surface area you have, the more sunlight you can get. So that might be an advantage to have, you know, shorter corallites that fuse together and create a more circular area.
One of the interesting things about this particular species of cropper is the way it reproduces. So it's an hermaphrodite. It's produced both egg and sperm abundance. By the time of the year, we'll spawn into the water column with a whole bunch of similar species.
Do you want to go biobox?
Whatever you want. Alright, I'm gonna go in 3 in the back.
It doesn't even matter if it goes in multiple boxes because it's so different.
Okay.
So you can put it right in the middle and wherever it falls.
It's so pairs there.
Yeah. You're getting lots of respect on YouTube and Facebook right now.
Actually, do you want to put it in 1 or 2?
I can put whatever you're comfortable with.
Well, I just feel like you're gonna, as soon as this close the lid. If we close the lid on this, is it going to break into all the bottles? That's okay?
Yeah.
Do you want us to put it in two?
Put it in two.
Yeah, sure.
Okay.
If it's easier, yeah.
We can get back. All right. If you're ready, I'm going to close one and open two. I'll just open two.
So we're just going to put it in a larger bio box.
Well,
All right, perfect.
Adam, that's brilliant.
Yeah.
Could have done better if you were doing it by hand.
Yeah, really. Okay, so we have hit the main targets of this dive, which is fantastic. So what we're going to do now, we have a few minutes left, so we're just going to zoom around a little bit in the shallows
52 degrees.
and see if something stands out.
Alright, coming up.
Yeah, another successful dive on the ROV Falkor.
We're going to look around a little bit while we're here because we got temperatures alright and see if anything jumps out at us.
Yeah, we're going to be up bottom here in like 5 minutes.
Yeah, see something really quick that we can do.
Tank bridge.
Eric, do you want to give Luke a little
Alright, recovering shortly.
Got some rope right there, so I'm going to come up.
So we've collected some of the expected genera at the top of the seamount, some really interesting stuff down a little deeper. So we have a lot to work with now to understand what the common species that occur on the seamount are. And of course we also have AUV data and the 5K video footage that's running on the ROV. So we'll have a lot of information for a lot of people to work with.
One other thing.
Alright, boys, we're going to get the deck team, everybody ready down here, and then let us know when you're ready to move on it.
Okay. Yeah, we're going to probably do a slide back in.
Yeah, because I noticed 101, so you can bring me in, Ben, or...
All right, guys, that'll be it for today for us. Thank you all for joining in. Stay tuned, though. For the next about 3 weeks, we're going to be diving almost every day. Different seamounts and reefs on the Coral Sea, and then the canyons in the Great Barrier Reef. So thanks all for your really interesting questions and comments, and we will see you again very soon.
Floats on surface.
Floats on surface.
1, 2, 3, 4, 5, 6. What's your currents?
Find out. Bridge control. Just looking for the current data.
Control bridge. Current. I'll get back to you in a second. Thanks. Control bridge, roughly 0.6 knots coming from northwest.
Copy that, 0.6 knots northwest.
Coming from the northwest? Pushing off to the starboard aft. Rails are awesome, by the way. How'd you guys say it was?
nothing crazy. I mean, good considering the time we had.
You can put me a little faster, Benner.
We are 129 from the vessel bend.
It's kind of nice.
Delta's at 95.
So now we have the process and the shell.
You got enough guys on there, Corey? You want me to come out?
I didn't know that.
You have enough guys on there? You want me to come out?
No, you're good.
Is there any chance?
Wait, do we have enough guys?